plenary sessions about drug. We learned many things. So just couple of slides about drug, a chemical substance that affect the biochemical processes of mind and body. And any chemical compound is used in the treatment or prevention of diseases or other abnormal conditions. And this drug can be categorized into two fundamental groups, like synthetic compounds or natural products or their derivatives. And a major portion of drugs are derived from nature. And we know aspirin, it was one of the first drug to be manufactured by the Bayer company in 1899, and it is mainly used for anti-inflammatory, and it is based on the bark of willow tree. We know medicinal plants play an important role in the primary healthcare, and about 80% of world population depend on these medicinal plants. And in history, if we see the history, we know more than nearly 3000 BC. In China, Emperor Shen Nung recorded the use of Ephrata species in Ma Hung. This is the traditional Chinese book about the uses of Ephrata species, and we know the Papaver somniferum, and from this opium tree, morphine, codeine, heroin were isolated. So these are the history of natural products and the uses of medicinal plants. There are more traditional user of medicinal plant like digitalis purpura, Atropa velladola for different purposes. In last century, we know penicillin was isolated from Penicillium notatum, and it is antibiotic. And in 20th century, many anti-cancer agents were isolated from medicinal plants. Like Taxol was isolated from Taxus brevifolia, podophylloctoxin, Podophyllum peltatum, and vinblastine and vincristin from Catharanthus roses. So these are some examples of success of natural product chemistry. Other than plant materials, marine organisms also play important role for producing secondary metabolites, and this slide shows different kind of invertebrate diversity, and all these natural sources can produce different kind of chemical compound, and this chemical diversity can give a biological diversity as well. So the natural product can play important role for combating different kind of diseases. And there are some records that more than 1,000 small molecules were approved as drugs between 1981 to 2010, and another 299 drugs were derived from natural products, and more than 177 synthetic structures were also derived directly from natural products, and more than 146 synthetic structures are modeled based on natural products. So natural products is very important for drug discovery. This is also full information. Over half of the world's 25 top selling pharmaceuticals owe their origins to nature. You see many medicinal plants and many active pharmaceutical drugs were isolated from these medicinal plants. To work on natural products, we have to follow some strategy like selection of plant materials, extraction of plant materials, isolation of secondary metabolites, and purification, structure determination, and biological activity. We have to focus on which particular activity we would like to carry on. In Bangladesh, we have many medicinal plants, and more than 5,000 floral species exist in our country, and more than 1,000 are medicinal plants, and out of them, nearly 800 medicinal plants have been listed so far. And our country, actually, diabetic patients are prevalent, and this is diabetics is a global pandemic, and we were carrying out research on anti-diabetic medicinal plants for a long time, and from this figure, we can see that diabetic patients are increasing globally day by day, and it is predicted that by 2025, the diabetic patient can be more than 438 million. And it is recorded in Bangladesh in 2011, nearly 8 million people are suffering from diabetes, and it is estimated by 2030, 16 million people will be affected by diabetes, and globally rank for Bangladesh will be 5th. So many people will be suffering from diabetes diseases. So we are looking for active medicinal plants, and its principle from diabetic medicinal plants. As we know, plants play an important role in treating diabetics, not only Asia, it can be in Africa too, and approximately 800 plants are used in folk treatment of diabetes according to ethnobotanical information. It is mentioned in this review paper. In Asia, if we compare different regions, in South Asia, India, Bangladesh, Nepal, nearly 36 anti-diabetic plants are available. You see from these slides, the different picture of different anti-diabetic medicinal plants like Bauhinia variegata, Coccinea indica, Gymnema sylvestre, Amada salicornica, Panax ginseng, Memorica gigantea, Tigulea ferropunum grassicum, Eurocoma gingivar, Morus alba, Taraxacum officinellum. So all these are different kind of medicinal plants are being used for the management of diabetics in Bangladesh and some part of India. There are some medicinal plants are also being used as antiviral, like Gingivar officinea, Justic Adatoda, Osimum basilicum, Solam sanctocarpum, Albizia lebic, Piper longterm, and Phyllanthus emblica. So these are examples of some plants which are also being used as antiviral plants. As I told, we are working on anti-diabetic medicinal plants, and we have worked with more than 90 plants for their hypo or anti-hyperglycemic properties, and these are from Bangladesh, Pakistan, some plants from Cameroon and other countries. And Gizipha rugosa, this is one of the plant, usually tribal people. We have some tribal people, and they are rich with different kind of plants, and they use this Gizipha rugosa for the management of diabetes. And we isolated several compounds like luteol, betulin, betanololdehyde, betonoic acid, and a flavonoid from Gizipha rugosa. Vetiver religiosa, this plant is also being used for the management of diabetics in Bangladesh, India, and Nepal. And we have isolated several phenolic compounds like paracomaric acid, paramethylsuccinic acid, ferulic acid, and a flavonoid from these plants. Omorica chayantia is all-known medicinal plants, and we work tremendously for this plant and its fruit. And actually, this is very active against diabetics, but we're not able to isolate many compounds. Only two steroid glycosides were isolated from these plants. Scoparia dulcis, this plant is used for the management of diabetics in Nepal, and the aerial part of the plant showed anti-hyperglycemic effect at different model rate. We isolated several new flavonoids from this plant, and some of these flavonoids showed very significant activity as antioxidant. Moria coinage, actually these leaves of this plant is also used as a vegetable, and this plant is widely distributed in Bangladesh, India, Nepal, and Sri Lanka. And again, the leaves of Muraya coinage is also used for the management of diabetes, and we isolated several flavonoids from this plant. Recently, we isolated a novel steroid glycoside with actually a steroid with 4 glucose molecule from Corypha tylia, and this is isolated. This plant is actually very rare in our country, and we're also trying to work with this more with these plant materials so that we can carry out some biological activity. One of our PhD students is working with Avorhoa bilimbi, and you see the bilimbi tree and with fruits. And in our country, some local people use this plant without any scientific evidence locally for the management of diabetics. So we started work with this plant, and Mijan isolated several compounds, and 2 of them are very known compound, one long chain alcohol and beta citoestrol, and he also carried out the phenolic content of this plant in different extract, and he found DCM extract contain highest amount of total phenolic content, and again DCM extract contain highest amount of flavonoid content out of other 2 hexane and methanol extract, and total oxygen antioxidant capacity again in DCM extract. So it might be DCM extract contain more phenolic type compounds. And he also carried out cytotoxicity assay against halacel, but none of his extracts were found to contain any cytotoxicity. Another students, Nazrul Islam, he is working with Phyllanthus niruri. Actually, these plants from literature, we found that it is also used as antiviral activity, and due to COVID-19 situation, we have planned to carry out with this plan for COVID-19, and we are developing some laboratory work. But again, with our facility, he tried to determine the total phenolic content, as gallic acid equivalent, and he found that ethyl acetate out of different fraction contain highest amount of total phenolic compound, and total flavonoid content was highest in ethanol extract, and total antioxidant capacity in ethyl acetate extract again. He also carried out cytotoxicity assay, but none of his extracts were found to contain any cytotoxicity. But some of 80% ethanol extract was showed weak activity against 2 gram positive and gram negative bacteria strain. Another PhD student, Hafizur Rahman, is at this moment working on Pseudium guazaba, locally known as piara, and this is very popular fruit in our country. Even we eat this fruit while we work outside or street. And recently, from literature, we found that this fruit, Pseudium guazaba, contain medicinal properties, and he was working on antidiabetic activities of this fruit and isolating compounds, and he developed for his work a diabetic model rate. You see from right side the picture, 2 days old pups, red, long even red, were used to induce streptozotocin injection, and after 3 months, when these states are little bit adult, he used for his experiment. And for his experiment, he categorized 4 groups. Group 1 only red with normal water feeding, then streptozotocin induced group, and again he used positive control by drug glycoside, and another is with his plant extract, and he was looking for the activity of this plant comparing with all other groups. And these are the biochemical parameter. Serum glucose was measured by glucose oxidase method. Serum insulin was measured by ELISA. Serum lipid was measured by enzymatic calorimetric method, and liver glycogen content was measured by anthrone sulfuric acid reagent method. And this slide is the chronic effect of the aqueous extract of the pear leaves on fasted serum glucose, and you can see that in the first one, the 0 day, when the extract was induced, then 14 days result and 28 days result. So these are the actually standard protocol from our previous experiment or developed method, as we are working for many days. And this Pseudium guazaba showed very good activity fasting serum glucose level from starting to after 28 days. And chronic effect of this extract on lipid profile, you see this bar chart, triglyceride from 0 day and after 28 for 4 different groups. And for extract, we see that the cholesterol level is decreased after 28. So it is also very good result from this extract. In terms of HDL and LDL, again, out of 4 different groups, extracts showed very good activity to lower LDL. And you see when it was on 0 day, the level was very high, but after 28 days, it's lowered significantly. And chronic effect on serum insulin type 2 diabetic model rate, and we see that this extract again showed very significant results after 28 days. And liver glycogens level, and this extract increased liver glycogen compared to other groups. So from his study, we can conclude that Pseudium guajava leaves possess anti-diabetic activity by stimulating insulin secretion, improving lipidemic status, and enhancing glycogenic in the liver. But still, we're working with these plants. Now, I will just share a couple of slides. I don't know how much time do I have, but just as you know, due to medicinal plants in this subcontinent, Ayurvedic medicine is very popular, and there are many companies in Bangladesh who are working under Ayurveda, and this is government recognized company. More than 220 Ayurvedic and more than 23 herbal companies are available, and there are many products from Ayurvedic, and Hamdard, this is one of the company. They claim themselves the pioneer leader of natural medicine in Bangladesh, and they have different products, and one of the product is Sincarra, and it provides energy and stamina. Actually, this Sincarra is mainly made of different kind of medicinal plants, although in this slide it is written in our local name, but you see different kind of medicinal plants for this Sincarra. Kobe is made from ginkgo biloba 16 milligram capsule, and it is also very popular in Bangladesh for boosting up memory and improves blood circulation. Zinton, it is from 10x ginseng 500 milligram capsule, and it acts as an antioxidant, adaptogenic, and increased energy and stamina. Safi, this is also a herbal product, and it is used for blood purifier, detoxifier, anti-aging, and antioxidant. Chayanbash Prosh, this is again Ayurvedic tonic, and it's actually made of different medicinal plants as Phlanthus imblica. I showed a picture of this plant before. It is Vinivera, Adatota vesica, and some other plants. And tablet products is used for weakness of vital organ and nervous debility and sexual debility. And you see the ingredient of this tablet, and it is actually a mixture of different medicinal plants. So from all this actually information, we can say that plants and of course medicinal plants play important role for combating different diseases, and they can produce bioactive secondary metabolites, and many plants have been used in discovery drug for the treating with diabetic patient. Again, we need to focus for isolation and identification of active constituents from anti-diabetic medicinal plants. Still, we have to work more on it. I'm very grateful to my colleagues, Professor Moshiuddin, Department of Chemistry, Professor Nilupar Nahar, Professor Begum Rokeya. Actually, she is expert for anti-diabetic work. Professor Marcel Jesper, one of my PhD supervisor from UK, Professor Satyajit Sarkar, Professor Paul Kong. As I mentioned, we received research fund from different organizations like IFS, OPCW, ISP I mentioned earlier, and locally from Ministry of Education, University Grant Commission. So thank you very much for your patience.
Thank you very much, Muhammad. That was a great presentation. I think I speak for everyone when I say that it's impressive the scope of work that is being done at your research group. What I'd like to do, I know we have about 3 or 4 minutes of the time. What I'd like to do is I'd like to open the floor for maybe just 1 or 2 questions from the members before we move to the next part of this session. So members, you have 2 options, as you probably have realized. One is you can raise your hand and I can ask you then to unmute and ask your question, or you can type a question in the chat, which can then be answered as we go along. So I'd like to welcome 1 or 2 questions, and as I wait for that, probably I can start with 1 question. Now, I noticed from some of your work that not only do you look at activities in extracts, but you also put in quite a bit of effort to isolate pure compounds and characterize pure compounds. So my question to you is, have you had any pure hit compounds that you've done some additional chemistry on to try and optimize the activity or optimize some of the physical chemical properties? Because I think that would be the next logical step after identifying the pure compounds. Have you done so?
Yes, actually, we did some, we isolated several alkaloid from Murray Koinenji, and their isolation amount was quite high. We carried out, but we did not mention here. Still, we are working on those isolated compounds. We haven't finished yet.
Okay, all right. Because that is encouraging to hear, because you find that in many research groups, once they isolate compounds, they leave it at that. They do not follow up. And I know you're a professor in chemistry, so I would expect that they would also want to do some additional organic chemistry on that. I don't know whether we have any questions on the chat. If not, yes, I don't see any questions, and I know we are over time. I do not want to extend too much further. So mine is just to thank you, Professor Muhammad Shoaib, for your excellent presentation and for being our lead speaker, for making time to be with us in this session. And if you'll allow me, then I'd like to move to the next part of this presentations. Yes.
Okay. Thank you very much. Thank you, everybody, for being with this presentation. Okay.
Excellent.
Yes.
Excellent. You're more than welcome. Members, what we'd want to do now is move on to the next part of this session. Like I said, this particular session A2 is on natural products, and it's divided into 3 parts. The first was our lead speaker, who's just spoken. That was Professor Mohammed Shoaib. Our next part, we have 4 speakers who are to speak for 10 minutes each maximum, one after the other, after which we'll have a short question and answer session where each of the 4 can field any questions for them. As I said previously, each of the presenters will share their screen and take us through their slides. So looking at the schedule, I think we are beginning with Mamosheledi Motibe. I can see you are present. Mamosheledi, are you there?
Yes, I'm here.
I hope I'm pronouncing your name correctly.
Yes, I'm here. Thank you.
All right, please go ahead and share your screen. As we do that, Ursula, if you're there, you're on standby, you'll be the next speaker. I see, if I can just look quickly down.
Yes, I'm here.
Yes, all right, so you'll be coming on after Mamosheledi. Mariam, are you there? You're our 3rd speaker. I hope Mariam will be there. If she's not, I hope she'll have joined us by then, after which we have John Monjoria as our 4th speaker. So, yes, I see Mamosheledi has her slides up. Welcome. We are looking forward to your presentation. You have 10 minutes.
Thank you very much, Chairperson. I'm trying to get my presentation in show mode. However, it looks like it's not getting there. I lose my time. Thank you very much.
You could press F5 on your computer.
Okay, let's see. Before I share the screen, okay, let me unshare and then share afresh. Okay, close share, stop share. Sorry about that. Then I'll get it in show mode and then share.
As you do that, I'll encourage members as we go through each of these presentations to take note of any questions that you might have. You can either note them down somewhere and ask them at the end of the session, or you can actually just type them into the chat. I'll be keeping an eye on that, and we can then have all the questions quickly when we get to the question and answer session. So just take note of your questions. You'll have an opportunity after the 4 speakers to ask a question to any of the 4 presenters.
Thank you very much, Chairperson. Good afternoon, ladies and gentlemen. My presentation is on the in vitro inhibition and stimulation of receptor and non-receptor based platelet activity in diabetes mellitus by commercial.
Mama Sheledi, maybe I can interrupt you. We do not see your slides.
Oh, wow. Okay. Let me do this. In show mode.
You need to share your screen.
Okay, I will share my screen. I will share. Is it visible now?
Summing up, there we go.
Okay, the challenge is that I once I start sharing, I cannot get it in show mode, in slideshow mode. So I will just proceed as is.
Just proceed as is.
Okay, thank you very much. As I was saying, my presentation is on the in vitro inhibition and stimulation of receptor and non-receptor based platelet activity in diabetes mellitus by commercial herbal mixtures used as African traditional medicines. Commercial herbal medicines or herbal preparations or herbal products that are used as traditional medicines have gained wide popularity, more so now than ever before in the face of the pandemic. These herbal mixtures are advertised as immune boosters, and although they are presented in nicely prepared modern packaging and presentations, they are efficacy, safety, and toxicity profiles are largely unknown, and we also don't know their effect on different physiological systems as well as in different pathological conditions. It is in the nature of people to seek health in various ways. So these commercial herbal mixtures then might be used by patients with chronic diseases, such as those with diabetes mellitus, as self-medication. And therefore, it's reasonable to be concerned about the potential impact on the outcomes of this disease management. One of the key elements of the management of diabetes mellitus is the control of platelet activity, because we know that platelets in diabetes mellitus are hyperactive or are hyperreactive, and they are associated with development of complications, particularly cardiovascular complications. Therefore, this study then sought to investigate the effects of select few herbal mixtures, commercial herbal mixtures, on platelets in diabetes mellitus, which is under control, as well as to elucidate on the mechanisms by which those effects are exerted. We performed a bioluminescence assay on platelets that were extracted from blood samples of consenting type 2 diabetes patients who were well controlled on their conventional therapy. And these patients were not on any antiplatelet therapy. They were also not on any self-medication for at least 14 days before the date of collection of the blood samples. The herbal mixtures that we selected were we bought in a pharmacy in Pretoria and South Africa. And at the time of the study, these were some of the commonly found commercial herbal products in various retail outlets in the area, in and around Pretoria. For the experimental procedure, we prepared 4 serial dilutions of each of the herbal mixtures. So for the experiment, for each herbal mixture we had 5 test standard or test solutions for investigation. In brief, the luminescence procedure, platelets were incubated with the herbal mixtures for about 37 degrees and 15 minutes. And at the end of the incubation, the platelets were activated with either one of the 2 agonists, either PMA or FMLP in short. Then their luminescence activity was measured in a luminometer. The luminescence readings then were recorded and then were used to calculate the percentage inhibitions using the formula that is displayed here, which takes into consideration the luminescence activity of the control standard as well as those of the test samples. We then graded the percentage inhibitions as moderate, as weak, moderate, and potent if they were above 80%. Now, PMA and FMLP are both agonists on the NADPH oxidase system, which results in production and release of reactive oxygen species. However, PMA directly activates protein kinase C, which activates this enzyme system, whereas FMLP actually binds to a membrane-bound G-protein coupled receptor, stimulating or initiating a cascade of reactions that result in activation of protein kinase C and activation of the NADPH oxidase system. I've got this diagram. These 2, the one on the left is a simplified version that shows the difference in the mechanism of action between PMA and FMLP, with PMA being a direct activator of PKC and FMLP binding to a membrane-bound receptor. And what's important is that the NADPH oxidase system is very important for the functioning of platelets. So I've got this picture obtained from Fuentes and Associate that shows the many processes that are mediated by the NADPH oxidase system and the reactive oxygen species that are released once the enzyme system is activated. So with the results then, in summary, we saw that there was in vitro inhibition as well as stimulation of both PMA and FMLP mediated activities after exposure to the herbal mixtures. Now, with the time-based effect, the time-based inhibition as well as stimulation of the platelets were non-systematic. They varied across the different herbal mixtures as well as across the different concentrations within each one of the herbal medicines. Generally, though, across all the herbal mixtures, the highest concentration standards had inhibition effects on all the platelet via PMA as well as via FMLP activation. And across most of these herbal mixtures, then the lower concentration standards had stimulation effect. This effect also varied according to the herbal mixtures as well as according to the different concentrations. When we looked at the average percentage inhibitions for 60 minutes that were calculated for 60 minutes, the magnitude of the FMLP mediated inhibition effects were all higher than the PMA mediated effect. However, the stimulation effects on the platelets were variable, and the levels ranged from the weak to potent across the herbal mixtures as well as across the different concentrations. I've got these 2 charts here to show the expression of the results as I described for herbal mixture number 1, which clearly shows the highest concentration standard that it actually exerted inhibition effects on both PMA and FMLP mediated platelet activity, whereas the lower concentration standards showed stimulation effects. And in contrast, the herbal mixture number 6 was different in that the 3 higher concentration standards had inhibition effect on FMLP-mediated platelet activities. We presented the results in a table for the average percentage inhibitions over 60 minutes, and the values presented in red show where there were inhibition effects, and those presented in black show the stimulation effect as evidenced by the negative sign also. The results presented in bold are where the stimulation effects ranged from moderate to potent effects in magnitude. So this was just to show how diverse the effects were of the herbal mixtures across across the 2 platelet activities mediated via FMLP or mediated via PMA. In discussion, we have seen in the results that exposure of diabetic mellitus platelets to higher concentrations of the herbal mixtures showed inhibition of platelet activity, which was evidenced by the reduced response of the platelets to agonists acting via the protein kinase C pathway, as well as via the formal peptide receptor pathway, which is a G protein coupled receptor pathway. Following exposure to lower concentrations of the standards, the platelets were more responsive to the agonist and had increased activity. The inhibition and stimulation of platelet activity actually translate to the antioxidant and the pro-oxidant behavior of the herbal medicines on the platelets. If we get back to the condition itself, diabetes mellitus, increased platelet activity is not a desirable state in the condition. So activation of the NADPH oxidase system and the subsequent release of reactive oxygen species would actually be an unwanted effect of these herbal medicines. Because activation of the NOx system, of this enzyme system, actually mediates processes such as adhesion, recruitment and aggregation of platelets, all of which would lead to coagulation and thrombosis. They would also lead to activation of platelets within the immune system and the inflammatory response. And with reduced activity though, the reduced activity of platelets might be a favorable thing in diabetes mellitus. However, generally we know that reduced activity of platelets is always associated with increased risk of bleeding. What is important that we found in literature is that inhibition of the NADPH oxidase, as reported by other researchers, carries less risk of bleeding. Hence, they recommended that inhibition of this enzyme system in platelets can actually serve as new targets for new anticoagulant agents that have a reduced risk of bleeding, and therefore that might present an effective antiplatelet strategy. In conclusion, the findings in this study suggest unpredictable effect of commercial herbal medicines that were studied, and these effects then would differ with the composition as well as with the concentrations of the herbal mixtures. The findings also suggest potential interactions between the herbal mixtures with conventional treatment options of diabetes mellitus, and the interactions can be at protein kinase C level, or they could be receptor-based interactions. The study highlights the need, therefore, for caution during management of diabetes mellitus for patients who might use or who may be using these herbal mixtures, because control of platelet activity is important for positive treatment outcomes of diabetes mellitus. The study for me highlighted the importance of understanding or of knowing and understanding how the various medicinal plants or how the different herbal medicines would affect platelet activities, and how important it is for us to understand the mechanisms by which they exert these effects, because effects on platelets carry lots of implications in health as well as in management of several various diseases. Thank you very much for your attention. These are my acknowledgements.
All right. Thank you very much, Mamosheledi, for that interesting presentation. I have a couple of questions that I'm sure I'm going to ask, and I'm sure members also have. It's been interesting. The contradictory, the conflicting rather results that you're presenting. So we'll definitely engage on that during the question and answer session. Thank you very much.
Okay, I'll stop sharing now. Thank you.
Yes, please. Yes, please. Thank you. Our next speaker is Ursula.
Yes.
I'm inviting you now to share your screen and prepare to present. As you do so, I would like to know whether Mariam Issa has joined us. Mariam Issa should be our third speaker. I see a Mariam, but I'm not sure whether it's the same Mariam Adenike. Might be a different Mariam to our speaker. John Monjoria. Seeing that those two speakers may be joining us or might be having issues with their connection, can I call on ICT to be ready to share their presentations after this coming presentation? Yes. So Evanson, can you confirm? Or Nelson? Anyway, I'll reach out to them directly so that we do not waste too much time. So Ursula, thank you. We can see your screen. You have 10 minutes. Welcome.
Thank you so much. So I'm going to present our result on Carissa edulis estrade, a medicinal plant of Benin pharmacopoeia, induced potent endothelium-dependent relaxation of coronary artery rings involving predominantly nitric oxide. It's known that 17.9 million people die every year from cardiovascular diseases, and these represent 31% of global deaths. 9.4 million of these global deaths is due to hypertension. In Benin, cardiovascular diseases represent 14% of death. The incidence of cardiovascular diseases is increased with aging, which, as we can see here, is characterized by impaired cellular and organ function. For example, according to American Heart Association, the percentage of people with cardiovascular diseases increased with ages, and especially after the age of 40s. The prevalence of hypertension high in low and middle income countries, and these affect people in those countries not only because of the high population density, but also because of the weakness of health system. In Benin, hypertension represents 27.9. Aging is one factor that contributes to the development of hypertension, and 2/3 of population in Benin, which have over 50, is hypertensive. Health system in Africa is associated with a low performance of available preventive and curative service and higher cost of service in hospital. So population used traditional medicine because is more accessible and less expensive. Léum ginandra, Diodia scandens, and Carissa edulis are 3 plants used in Benin for the treatment of hypertension. Now the question is if the biological activity on the cardiovascular system of Carissa edulis, Diodia scandens, and Léum ginandra. The question is to clarify this activity of the plants. First, the aerial parts were drying grinding, and we did extraction with water and ethanol. And after that, we did we fractionated the extract with 4 different solvents. And then we did vascular reactivity. So the coronary artery were removed and cleaned of the conjunctive tissues. And then they were cut into rings with or without endothelium before being placed in organ chamber connected to amplifier. Tissue organ buds were provided with normal temperature oxygen and nutrients in the form of Krebs solution. The vascular reactivity was performed by exposing the rings to different vasoactive compounds and inhibitors. So as we can see here, Carissa edulis crude extract induced strong relaxation on rings with endothelium compared to rings without endothelium. And this endothelium-dependent relaxation is more pronounced than that of Diogenes candens and Cleomgynandra. So we continue the works on Carissa edulis extract. And we try to compare the relaxation of crude extracts with all other fraction. And as we can see here, the butanolic fraction was more active than all other fraction and also than the crude extracts. Here we did the kinetic of we try to compare the kinetic of Carissa edulis butanolic fraction and bradykinin. And as we can see here, Carissa edulis is more stable than bradykinin. So it causes sustained endothelium-dependent relaxation. Now, to characterize the mechanism by which the extract did this relaxation, we used different inhibitors like indomethacin, the inhibitors of prostanoid, LNA, the inhibitors of endothelial NO synthase, and both TRAM and UCL inhibitors of endothelial derived factor. So as we can see here, when we used the inhibitors of endothelial NO synthase, we can see that the relaxation was stopped for the crude extract and the butanolic fraction. So we conclude that Carissa edulis crude extract and its butanolic fraction induced relaxation involved predominantly NO production. Next, we try to see how Carissa edulis butanolic fraction can inhibit contractile response to KCl, calcium, U, and serotonin. And as we can see, in concentration dose dependent manner, Carissa edulis butanolic fraction can inhibit contraction to KCl, calcium, U, and serotonin. And this inhibition is mediated by the NO, the endothelial NO. So in conclusion, Carissa edulis leaves extract has a potent endothelium-dependent relaxin, is a potent endothelium-dependent relaxin natural product and an inhibitor of contractile response. Sees mostly to the endothelial formation of NO. The NO is an important endothelial factor controlling vascular and cardiac homeostasis. The vasoactive compounds of Carissa edulis leaves predominantly present in its butanolic fraction. Thus, Carica edulis leaves extract appears as an interesting natural product to control vascular tone and hence blood pressure. Thank you for your attention.
Oh, sorry, I was muted. Thank you very much, Ursula, for that interesting presentation. I am not sure whether our next two presenters are here. The last I checked, Mariam, Isa. I don't see Mariam Isa on our list, and John Monjori, I also haven't seen him on our list of participants since the morning session. So what I'd like to do, if you'll allow me, ladies and gentlemen, is to take questions, because what follows after those two presenters is a question and answer session for the would be 4 speakers, which was to begin at around 20 minutes to 3 o'clock local time here. I see we are just a few minutes short of that. So I'll open the floor for questions. As you probably have seen, there are 2 ways that you can ask your questions. You can either put your hand up and I can invite you to ask your question on the floor, or alternatively, you can type your questions on the chat and we can see whether we can lift them from there. So members, like I said before, you were to take note of the questions you wanted to ask our presenters. Right now we have 2 presenters. Feel free to ask questions to our lead speaker in case there were any that were pending, and we can proceed from there. So I have a few questions, but I would like to pick up a couple of questions that have appeared on the chat. So this question is for you, Mamo Sheledi. The question is, did you carry out acute toxicity on the herbal mixtures that you are working on? Did you carry out acute toxicity on the herbal preparations that you are working on? As you prepare your answer to that question, Ursula, there's a question for you. The question is, why didn't you carry out the effects of your extracts on lipid parameters, since they are also implicated in hypertension? Is it something that you considered? So why did you not carry out the effect of your extracts on lipid parameters, as they are also implicated in hypertension? You can begin preparing to answer that question. So Mama Sheledi, what was the answer to your first question? I invite you.
Thank you very much, Chairperson, for the question. I saw them on the chat. We didn't do acute toxicity studies on these herbal mixtures. We just performed this in vitro assay on platelets, and we actually exposed the platelets to the herbal mixtures immediately. They were extracted after the process of extraction. So we didn't actually perform any acute or any long-term toxicity studies on them.
Okay, is that something that you're probably considering to do? Are you still working on these mixtures, or you've moved on?
This showed us that there's potential for further studies on these. But as you know, animal studies require a lot of ethical considerations. But if we are well resourced, we might just continue with this and do in vivo studies to see how they relate with the in vitro studies.
Okay, so you basically saying you've put a pin on that. If you have the resources, it's something that you might consider.
Definitely.
Right. Thank you very much. Ursula, there was a question for you on why you didn't carry out the effects of your extract on lipid parameters. Would you like to respond?
Yes, thank you so much for this question. Yes, exactly. Lipids are implicated in hypertension, but for our work, as we just start So I think we will try to evaluate also lipid parameters. We will do it after.
Okay, and probably I can just slide my question in here. Now, in considering the mechanism of relaxation that you managed to elucidate, do we have any drugs on the market with equivalent mechanisms of action? Because that can allude to possible clinical applications of your findings. Any drugs that you, in your literature review, that have similar or comparable mechanisms?
Yes, there are many drugs that have similar effects because I have like other colleagues that works on 3 other plants used in Benin to treat hypertension, and as we compare the effect, it's the same. So yes, other drugs also have the same effect.
Exactly. So what is the next step for you in this research?
The next step will be to evaluate other parameters like lipid parameters. We also did an in vivo treatment, like we used rats and aged rats, and we tried to see if our SRAT will have effect on the cardiovascular system, and we have good result about it, but I don't put it in this presentation. So the next step for me will be to evaluate some parameters like lipid parameters and see if they are also implicated in this effect of the plant.
Okay, so what you're telling us is that we should be on the lookout for your publications in the near future, yes?
Exactly.
Good. All right. So back to you, Mamoshe Lady. There's another question. I really hope I'm pronouncing your name correctly. So there's someone who says there's a simple question for you, whether or not you studied what the plants in the herbal mixtures were. So what were the plants that are present in the herbal mixtures? And secondly, there's another question, a follow-up question, which is asking, can there be a reason why at low concentrations you have stimulation of platelet activation and contradictory results at a higher concentration? So someone is asking you to comment on the possible explanations for this. So 2 questions.
Thank you for the questions. You are pronouncing my name correctly. I'm just seeing how there's so many variable spellings of the name on the chat, but you can call me Mamza for short. The question about what are in the herbal mixtures, because these are commercial herbal products that we bought, we were relying on the label or what the manufacturer presented as the ingredients within this. So for an example, with the herbal mixture number 1, the label said it contained aloe, water, assorted herbs, and it also had HO2, which is hydrogen peroxide. And herbal mixture number 6 just said it contained natural herbs. We actually tried much later on the following year to try and do a TLC assay with some students for this, but we couldn't confirm the actual components or the plant compounds, but we do know that commonly plants contained polyphenols, there would be flavonoids, there would be alkaloids. So we actually, I don't have a direct answer to say definitely there's this in these compounds, but we took what is on the labels as what the components of the mixtures were. It's very interesting because, for example, herbal mixture number 2 also contained African potato, aloe ferox, African ginger, Sutherlandia, and wild olive. And some of them also now have preservatives, which brings in another element in terms of now the composition, because when you use these medicinal plants as they are as traditional medicines, you don't have the preservative in the mixtures. So it brings in another element to how we are now going to proceed forth if we are preparing traditional medicines for use by the general public. Because are you going to add preservatives to these and still maintain that these are traditional medicines as they were? Or how do we plan to ensure that they still remain viable without any chemical additives, without any synthetic additives to them? So that's in short what I can say relating to that question. And
okay. So before you answer the next question, I think you've alluded to some of the more pertinent questions, especially with regard to commercial herbal mixtures. More often than not, we are not really sure what is in those mixtures. And if we know the mixtures, we do not know the relative quantities. And we do not know whether there are additional components, constituents, and the reason for that. So this is an area which we can have quite a lengthy discussion of what should and shouldn't be done. So yes, you had another question?
And the other question says, can there be a reason why at low concentrations the extract stimulated platelet activation with these contradictory results? I am not sure about why there's these contradictory results. However, looking at the fact that we were taking platelets that were from patients who were already on treatment. So these are not platelets that are pure or in essence that are not exposed to Herbal. These are platelets that are already exposed to some medicines or some chemical agents. So I think there could be an element of interactions. Also, we don't know for a fact the components of the plant compounds in each of these mixtures as well as their concentrations. So that is why it's not easy to try and find a reason as to why they give us these contradictory results. One other interesting aspect with the contradictory results is that in an earlier study where we did the same procedure, when we took platelets from healthy volunteers or self-proclaiming healthy volunteers, we got totally opposite results in that all of these herbal mixtures actually had different levels of inhibition effect on the platelets. And those were platelets of people who do not have diabetes. So it's very interesting as to how platelets can be affected by these mixtures. And what it tells me is that until you know for a fact what compound you are testing, we cannot really attribute, properly their activities or their effect on the platelets. It might be a matter of availability of the receptors because of lower concentrations. It might be a matter of competition at the receptors. I don't know. There's no particular reason I can give for this kind of results. What they're telling me, honestly, is that there's a lot more to the herbal medicines when we take them or when people take them than it, meets the eye.
So there's a final aspect to this. What do you think? Now, this is generally your comment on the use of these products in diabetes management. Do you think, what are your comments based on your findings?
From my results, at higher concentrations, the platelets were inhibited, and this would be a favorable thing in diabetes in general. But then, how would you know that you are having the highest concentration possible or the highest dose possible of these medicines? Chances are, according to me, chances are in the body, the concentrations would be much lower than the highest concentration we had in the procedure. So I would really advise patients who are on treatment not to take these medicines, because if my hypothesis is right, in the body the concentration would be much lower, which means then it translates to activation of platelets, which might be an unwanted effect.
Right.
So I would really advise that when a patient is on treatment, rather stay away from any self-medication with herbal medicines because you don't know how they will affect the condition or how they will affect components such as platelets.
Right.
I would rather we avoid them.
Okay, so there seems to be quite a bit more work needed to be done there in that particular area, yes? All right,
Yes.
and then finally, I think this question is for you, Ursula. What is the potential, what are the potential predictors for the degree of antihypertensive activity apart from extract concentration? So question is asking you, what are the other possible predictors for the activity that you saw aside from possible concentration dependency? Ursula? Ursula, I think, I'm not sure that she's still with us. She probably has dropped off. That's one of the challenges with technology and all this, yes? So we'll put a pin on that. We can always revisit should she rejoin us. So ladies and gentlemen, if there are no other questions, I don't know whether anyone has put their hand up. I think we've handled all the questions for our two presenters. Mine is just to thank them for their excellent presentations and for the capable way that they've handled the questions. As usual, you can ask questions. Again, we'll have another question and answer session. But allow me to move this session forward. This is now about 10 minutes to 3 local time. I think we are doing well for time, despite the fact that two of our presenters seem to have had challenges. So I'd want to move to the third part of our session today, which is we have another set of 4 presenters. Let me first of all confirm that they're all here. Salisu, I think I saw you. Are you with us? Salisu, say hello. I can see you, Mohammed Higab Salisu. I can see you as one of the participants. I can see you already. Okay, you're already sharing your screen. Good. As you do that and prepare to present, we have Fatima Ismail Hassan. I think I've also seen you as one of our presenters, so as one of our participants. So I'm putting you on standby. You'll be presenting immediately after Salisu. Jawar Lamek, I don't think I've seen him or her.
Have been
almost
Salisu, all the
Salisu, if I can just interrupt you a little bit, there's a speaker that is giving us quite an echo. I think there's someone else who's also has audio in the room, the same room.
Yeah.
So if you can just deal with that. Sorry about that. All right.
So almost all parts of the plant have medicinal property. In Nigerian, tamarind is freely and readily available and has been reported to be one of the most rampant treatment for cold, fever, stomach disorders, diarrhea, jaundice, and as well as skin cleansers. It applies on inflammation mixed with salts as an ailment for rheumatoid and used for algal salt strokes. Next slide, please. It also relieves pains, reduce secondary bacterial infections, and promote healings. Other reports in the literature suggest that a specific part of the plant has been used in traditional medicine. The leaves are used to reduce inflammation, inflammatory swelling, treat tremor, tumors, and ringworms. Blood diseases, smallpox, ultimate, and other ocular diseases and eye ticks are also part of what some of the diseases that these leaves are being used for. Next slide. The flower part of the plants are used also to stimulate appetite and also used in the treatment and of urinary discharge, as well as bad odor due to perspirations. The fruit part of the plant is also used for laxative, tonic to the heart's ultimate antihelmintics, and also for healing wounds and fractures. The treatment of this part of plant also has also been used for bile disorders and also used in the stimulation of appetite. The bark of the plant has been extremely a property that has been used for the treatment of ulcer, liver complaints like jaundice, also used in the treatment of the diabetics mellitus. Next slide, please. Next slide. 13 distinct compounds, which include the linolenics and benzoate, have been also isolated from the leaves of this particular plant. The aims of this study is to, therefore, is aimed to evaluate the analytic activity of methanolic leaf extracts in Tamarazus indica plants in mice and oyster rats. Next slide, please. The specific objective of this study is to evaluate the quantitative and the qualitative phytochemicals of tamarins, of this particular extra actually, and to also to evaluate the acute toxicity profile of this extract, and to also to determine the analgesic property of this extract. Next slide. Methodology. Actually, experimental animals were used, as I said earlier on. We used adult albinos Swiss mice, and also the oyster rats of both sexes, ranging from 150 to 200 grams, and aged between 3 to 5 months old, were obtained from the National Institute for Pharmaceutical Research and Development, IDU, FCT Abuja. They were housed in the cage and they kept in a well-ventilated rooms under standard conditions of temperature and light in the animal house of Institute of Pharmaceutical Research and Development, IDU, in Abuja. Next slide. They were allowed to be acclimatized for laboratory conditions for 2 weeks prior to the study, feed on a commercial pelleted grower mash prepared in a pellets and given a water ablution for, except when fasting was necessary in the course of the study. All experimental were performed in accordance with the principles of laboratory animal care and then according to the NIPRI standard operating procedures. Next slide. Next slide. Collection of and identification of the plant material. The fresh leaves of the tea indica were collected from Geso in Korzo local government area of Kano State in Nigeria, and between the area of August 2018. The whole plant was identified and authenticated at the Iberian section of the National Institute for Pharmaceutical Research and Development in Idu in Abuja. The voucher specimen number as displayed in the screen was given and deposited in the Iberian section for future references. Next slide. Plant preparation of the methanolic leaf extract. The leaf of the tea indica plant was formed well fresh and air dried at the room temperature for 2 weeks. The dry leaves were actually reduced to a fine powder using a mortar and pestle. The powders of the leaves were extracted in the the rotary evaporator and the evaporated dry skins in the water baths at 45 degrees Celsius. The extract was also well stored in an airtight container and refrigerated for prior to be used for the experiments. Next slide. We carried out the method of quantitative determination of phytochemical of the extracts was carried out. The preliminary phytochemical screening analysis was carried out for the extracts to identify the presence or the absence of the organic metabolite according to the method described by Evans and Sophros, 1993, and the quantitative determination of the phytochemicals of the extracts also, like the alkaloid, the flavonoids, the tannins, the cyanogenic glycosides, the saponins, all were carried out according to the method described as displayed in the screen. Next slide. Also, acute toxicity study was also carried out by the method of Lux 1983, and also followed by OECD guidelines 425 for acute toxicity study, OECD guidelines 2001. And we also we're able to carry out analgesic activity of the plants through carrying out the method of septic induced riding test on the mice, and hot place induced pain in the mice also. And also we carried out a formalin induced hemmed pouch linking on the Wister rats. Next slide. Data analysis. The data analysis was the data was analyzed using a statistical software of SPSS version 20. The quantitative data were expressed as mean plus or standard error of mean. Significant differences between the mean were assessed using one way ANOVA at 75% level of significance. P less 0.05 was considered as significant. The Dunet post hoc test was carried out to determine where the level of significance lies. Next slide. Result and discussion. The plant extracts, the methanolic plant leaf extracts of T. indica yielded 19.9% weight by weight, and solid brown rich powder, which was water insoluble except when mixed with the target can as a suspending agent. The quantitative determination of the phytochemicals of the extract, the quantitative screening of the extracts revealed the presence of glycoside, flavonoid, saponins, alkaloids, reduced sugars, steroid, and terpenoids, as will be seen in the table 1. Next slide.
Salisu, just to note that you're done with your 10 minutes, yes? So you should be wrapping up now, yes?
As you can see, according to the table 1 here, present, the following, all these phytochemical constants, the plus indicates the presence of this phytochemical in this extract, while the negative indicates the absence of these phytochemicals in this extract. Next slide. Table 2 here shows the quantitative determination of the phytochemicals in 100 grams of the extract. And this result actually shows that the alkaloid happens to be the highest percentage yield in this present extract, followed by the flavonoid and the tannins. Next slide. Next slide. In the present study, the oral LD50 of the extracts was found to be greater than 500 milligram per kg in both mice and rats, suggesting that the extract is non-toxic when administered acutely. The test animals did not exhibit any visible change and survived beyond recommended durations of observations in up to 8,000 milligram per kg. Hence, the extract was safe up to 800 milligram per kg, as also confirmed by the OECD guidelines. Next slide. This table 3 actually showed the effect of the extract on acetic acid-induced Wistar riding in the animal and in the Wistar rat. And it shows that significant, there was a significant difference when compared to the normal saline group, that is the control. And it shows that this present phytochemicals that are present in this may have been responsible for the effects. That shows that the extract has actually shown to be potent in terms of the analgesic activities of the plants. Next slide. Next slide. The effect of the extract and the hot plate reactions also in the mice was also examined in this study, and we're able to see significant differences between the doses of the extracts that was administered to this mice as compared to the normal saline group. And that shows that the ability of the mice to attend to the hot plates has actually been indicated, and it shows that the extras has actually reduced the potency of this effect and in the mice. That also give an evidences that it has analgesic activity and presence in the plants. Next slide.
Salisu, I need you to give me one more slide as a maximum, yes? In the interest of
time. This is the final table that we have, that is the formalin-induced POC link test. And actually, it also shows that the doses that we administer to this flight shows significant differences as compared to the normal saline group. And it shows that this actually has an effect also on the Wistar rats. Next slide. In conclusions, the methanolic leaf extract of tea indica possesses phytochemical constituents that may be responsible for central and peripheral mediated analgesic activity. These findings will provide more scientific evidence for the ethanol medicinal uses of the tea indica leaves in the management of pain. Next slide. These are my references.
Okay, thank you very much, Salisu, for your presentation. I think you've exceeded time by about 5 minutes. Fatima.
Thank you very much.
All right, Fatima Ismail, can you prepare to share your screen so that we can? Fatima Ismail, are you with us?
Yes, I am here.
Okay, kindly share your screen and prepare to present. Salisu, please mute.
I'm finding it difficult to share my screen.
Fatima, are you having an issue sharing your screen? Evanson, can you confirm whether or not you have Fatima's slides that we can use the same approach for her as we did for Salisu? Evanson? Okay. As we try to sort out Fatima again, in the interest of time, Jawar, I don't believe has joined us.
We have the presentation for Fatima. We're sharing it in a moment.
Okay, please do. Fatima, your presentation will be shared on your behalf, and you can just take us through in the same manner as Salisu did, yes? So you can stop sharing your screen now. Excellent. Those are the challenges that we expect to face, but we shall power through. Fatima, welcome. You have 10 minutes, and please adhere to the 10 minutes, yes? Fatima. Fatima, you need to unmute. We cannot hear you.
Okay. I'm sorry about that. Good
All right,
afternoon,
10
all. I'm presenting on the antioxidant potential of the methanol leaf extract of Dalbergia saxatilis using the cotton pellet-induced granuloma model in albino rats. These are my co-investigators. Is the next slide. This is my outline. The next slide, please. Majority of the population or majority of the people in Africa, if I can use that, use medicinal plants due to accessibility and availability. Oxidative stress is usually a serious problem in terms of many diseases, and it usually results from excess production of reactive oxygen species that overwhelms the activity of our antioxidant defense. The first line of antioxidant defense is usually our superoxide dismutase, which I'll talk about them one by one. Next slide, please. Sorry, I don't want to dwell on the introduction. Antioxidants are molecules that inhibit the oxidation of other molecules, and for the course of our own research, we talked about reduced glutathione, superoxide dismutases, catalase, and also MDA, melon the aldehyde, which is MDA. Sorry, the spelling is wrong here. And reduced glutathione usually protects the mitochondria, and it also improves the mitochondrial function, while superoxide usually counteract the effect of oxidative stress markers, which is free radicals, by converting them to hydrogen peroxide and oxygen molecule, and it acts in conjunction with catalase, which also converts the hydrogen peroxide to water, and oxygen molecule in 2 steps. While MDA, which is malondialdehyde, is the product oxidation, and because of our work. Next slide, please. Next slide, please. Okay, this is just a diagram that shows the effect of the endogenous antioxidants. We also have the exogenous ones. In our case, we used ascorbic acid as our positive control. Next slide, please. This Dilbagia saxatilis has been used traditionally in the treatment of many inflammatory and infectious diseases such as toothache, skin lesions, and smallpox. And also searches have been carried out, which shows the effect, the promising effect of this plant extract on uterine smooth muscles, on epilepsy, on pain, inflammation, and pyrexia. Next slide, please. Though I don't have a slide on aim, but the aim of the study was to determine the effect of this extract on oxidative stress markers. We have our plant adult Vista rats, which were obtained from the animal house facility of the Department of Pharmacology and Therapeutics. I also used our chemicals and drugs, and these are our equipments. The next slide, please. The plant was collected in Giwa, local government area of Kaduna State, and it was identified and authenticated by a taxonomist, and a voucher specimen number was deposited for further reference. Our plant was dried and macerated using standard procedure as described by Tris and Evans, and it was also dried. It was dried under reduced and controlled pressure and temperature until the dried extract was obtained and then kept in tight containers for tight container for further use. Next slide, please. We induce stress by using this cotton pellet granuloma method, where the animals were shaved and anesthetized. Then they were cotton pellets were made from cotton weighing 100 milligram each and were implanted in the groin area of the rats that were pre-shaved before the procedure. And before then, 20 minutes before the implantation, the extract and the vitamin C treatment were administered. And it continued for 9 days until on the 10th day, the animals were sacrificed. Before sacrifice, the pellets were removed. They were dried under 60 degrees centigrade and until a constant weight was achieved, and then weighed, while blood samples were collected in plain bottles for the determination of the oxidative stress biomarkers. Next slide, please. MDA, which is malondialdehyde, SOD, catalase, and GSH were determined spectrophotometrically according to manufacturers' specifications. Next slide, please. Our results were expressed as mean plus or minus standard error of mean and presented as graphs. And our data were analyzed using one-way analysis of variance, followed by Turkey post-hoc test for multiple comparison. Our results were considered significant at p less than or equal to 0.05. The next slide, please. This was our experimental design. We had 5 groups of 6 rats each. And the treatments were group 1 normal control, which received distilled water. Then our positive control received vitamin C, 250 milligram per kg. And then group 3, 4, and 5 received graded doses of our extract, which were 250, 500, and 1000 milligram per kg, respectively. And treatments were done per oral. Next slide, please. So our yield was 30.6% weight by weight, which is quite on the high side. The next slide, please. Using this cotton pellet induced granuloma method, we didn't observe any significant difference in the inhibition, though there was inhibition, but very low, very light, even in the vitamin C group. Though vitamin C is an antioxidant, so we didn't expect it to show a very promising activity on cotton pellet induced granuloma method, which is an inflammatory model. And same goes for our extracts, that all the groups, there were no significant inhibition, though there was, but not much. Next slide, please. Next slide, please. I can't see it, maybe my network. Okay, sorry, the next slide is MDA, I missed it, my network was bad. So, but this SOD, our MDA, which is a product of lipid peroxidation, was inhibited when compared to the normal distilled water group. We can see that there was a much increased red bar, which shows that there was lipid peroxidation, that is oxidative degeneration. But luckily, our plant extract was able to reduce it. That's the highest dose group tested compared to vitamin C, was similar, which shows that our plant, our extract, has the potential for reducing lipid peroxidation by reducing the levels of malondialdehyde. Next slide, please. Superoxide dismutase, which I said is very important in oxidative stress. It helps break down potential harmful oxygen molecules in cells. It converts these superoxide free radicals into hydrogen peroxide and oxygen molecules. And it acts in conjunction, I said earlier, with catalyst, while catalyst reduces this hydrogen peroxide to water and oxygen molecules. Our plant, as the highest dose tested, was also significant, though not much, but it was significant compared to our distilled water group, which shows that our extract is capable, at the highest dose tested, capable of improving oxidative stress by increasing the levels of superoxide dismutase. Next slide, please. Glutathione, which is involved in many situation, nutrient metabolism, antioxidant defense, and also regulation of cellular metabolic function, is also important in minimizing lipid peroxidation of cellular membranes. And our extract was able to increase it. Though this time around it was increased, it was more significant at the lowest dose, which shows that increase in our dosing of our extract will not increase the activity of, will not increase the level of reduced glutathione. Next slide, please. Next slide, please. Okay, catalyst, in this case, there was no significant increase, though there was increase, as you can see from the bar chart, but not statistically significant. That means it's possible our plant was able to increase the level of catalyst, but not significant at this level. But as I said, it works in conjunction with SOD, and we saw significant increase in SOD levels. It's possible that our plant, maybe using when we carry out further studies, we can see its effects clearly. The next slide, please. In conclusion, this study suggests that our methanol leaf extract was able to reduce the level of MDA and also increase the level of the antioxidants, which may show its potential for antioxidant properties. Next slide, please. So these are my references. These are my references. Thank you for listening.
Thank you very much, Fatima, for your presentation, and also for keeping time, exactly 10 minutes. We are grateful for that. Just a reminder to all members, remember that we shall be having a question and answer session after our last presenters. So please either take a note of your questions and then you'll raise your hand at the appropriate time and ask your question, or feel free to type your question into the chat and we shall be going through those questions and each of the presenters will be able to answer them. So the time for question and answer is coming, so please do not lose track of any questions that you might have. Thank you very much, Fatima, for that.
Thank you, Sean.
Okay, I see that I'm not sure whether we have Jawar Lamek. I don't think he's joined us. I don't see him.
Yes, yes, I'm here.
Oh, you're here. You're finally here.
Good afternoon, everybody.
Yes, welcome. So
Thank you.
please share your screen if you can.
Okay.
Yes, you'll have exactly 10 minutes to make your presentation. In the meantime, Abey, Prakash, Mishra, you'll be presenting after Jawar, okay. So just be on standby. Jawar, 10 minutes exactly, yes?
Okay, thank you.
All right,
good morning, everybody. My presentation is about pharmacological activities of Berberis vulgaris against trace element intoxication. That is the PB, the lead using lead acetate mice model. I'm Jawhar Lamish. I'm from the Faculty of Medicine and Pharmacy of Tangier, Morocco. So lead is a dangerous systemic toxic metal that is included in lots of technological activities. So it gives a wide distribution in the environment and an increment environmental contamination for human. The exposure in human cures mainly through intake of lead contamination food, water, dust, and the degradation of plants. Actually, the detection and prevention of lead intoxication is still a preeminent international public health concern. High level of lead chronic intoxication shows that it is a cumulative and multi-target toxicant that generates EROS, reactive oxygen species, and depletes the cellular antioxidant capacity that cause finally damages in membrane, DNA, proteins, and finally destroys the lots of tissues. But the low-level chronic PB intoxication that causes Saturnism, as we see in this image from the prehistorical air history, that is now these mechanisms are partially, not fully elucidated in kidneys and liver, and are crucial for PB assessment and risk management in humans. Actually, there is an increasing interest in according that is accorded to polyphenols as strong antioxidant molecules in the fight against toxic metals, especially lead, because they are safe, natural, accessible, low cost for that we can use it as a prevention method for poorer population and as a prevention, and it can have a potential new active molecules against light or low level of chronic PB intoxication. So the aim of our work was to elucidate the oxidative mechanisms of low-level chronic lead nephrotoxicity and hepatotoxicity, and to investigate the effect of Berberis vulgaris L, BV, on this nephrotoxicity and hepatotoxicity, and to do chemical screening on polyphenols contents in BV. So the classification of Berberis, it belongs to the Berberidaceae family, okay, and the common barberry. And the parts used in our study was the stem bark of the plants. Material and methods. So we used 72 mice syrups that was gavaged that was divided into different groups. The normal control group received distilled water for 40 days. The toxic control group the same time they received the PBAC, lead acetate, for 40 days, and the treated groups received PBAC for 10 days, followed by 30 days of gavage of lyophilized aqueous extracts of Berberis vulgaris, co-administrated with PBAC. BV was administered in 4 doses, 25, 50, 100, and 150. So each group contained six mice. The chemical or parameters that was evaluated was the body burden, intracellular concentration of lead in kidney and liver, and biochemical parameters determining the renal function, creatinine, urea, and uric acid, and hepatic function, ALAT, ASAT, total bilirubin, and total cholesterol. The parameters of antioxidant activities in kidney and liver was MDA, PCO, GSH, GPX, the antioxidant enzymes, GPX, catalase, and SOD. We did an anatomopathological visualization assessment in kidney and liver, and finally the BV lyophilized aqueous extracts. We do phytochemical screening for searching the content of total polyphenols, flavonoid, flavonol, and condensed tannin. So the results, there's a lot of results to be fast. So the PBAC caused an elevation, an increase in the lead body burden in blood and in kidney and liver, and alteration in all these biochemical parameters in liver and kidney, and Berberis vulgaris, BV, the extracts of BV could reduce this alteration and restored the value of these parameters to values that are near to the normal, or sometimes to the normal values. So for the lead body burden, this restoration was in blood, as we see. So PBAC elevates the lead body burden in blood, and the BV could restore this value to a normal value from the dose of 100 milligram per kilogram body weight. In the kidney, from the value of 50 milligram, in the liver from the value of 100 milligram. In serum creatinine, urea, and uric acid was all restored at normal value from the dose of 150 in kidney. In the liver, ALAT and total bilirubin was restored at the dose of 150, and ASAT and the total cholesterol from the dose of 100 milligram per kilogram body weight of BV. Renal antioxidant parameters in kidney, PCO and GSH was restored from the dose of 150. Of course, MDA and PCO are lowered by BV. MDA from the dose of 100 and PCO 150, and GSH was increased by the dose of 150. The results can become significant. In the liver, MDA from the dose of 150, PCO the dose of 100, GSH was elevated from the dose of 50 milligram. In the liver, in the kidney, GPX and catalase activities were increased from the dose of 100 and SOD 150. In liver, the GPX and SOD activities were increased from the dose of 150. The result is significant. And for catalase, from the dose of 100. So the histopathological assessments showed that there is hypercellularity in the toxic groups that disappeared from the dose of 150. In liver, we had some binucleation, some focal necrosis, and polynuclear infiltration that was restored to normal structure of the tissue from dose of 100 milligram per kilogram. The polyphenol content was elevated, especially flavonoids that represent about 70% of the polyphenols, especially flavonols and condensed tannins. As a conclusion, PB extracts that contained a high content of flavonoids, especially flavonols and condensed tannins, it fights the side effects of PB by chelating PB in lead in blood and in the tissues, and potentiating the antioxidant system that can, as a benefit, increase GSH and the activity of antioxidant enzyme GPX, SOD, and catalase, and by this way decreasing the generation of erox. That was manifested by the decrease in MDA, products of lipid peroxidation, and the PCO, protein carbonyl, products of protein oxidation. Finally, the structure, the alterations in structure and the function of kidney and livers were corrected in normal structure and function of kidneys and liver. Thank you for your attention.
Thank you very much, Jawar, for excellent presentation and some interesting results there that we might want to revisit and ask a few questions about. Very, very interesting findings, to be honest. Although we've gone about 3 minutes over time, but we're not doing too badly in terms of time. We have one more presentation. Ladies and gentlemen, I would like to remind you to keep track of any questions you might have. The question and answer session is after the next presentation. You can type your questions into the chat, or you can put up your hands at the appropriate time and we'll be enabled to ask your questions. Yes. So thank you very much, Jawar. You can stop sharing your screen now. And we move to our next presenter, who's the last presenter for this session. This is Abey Prakash Mishra. I know you're with us. So prepare to share your screen, Mishra. And
yeah, Dr. Eric, hello.
Yes.
I'm here.
Yes.
Okay,
so
let me start my presentation. Another one is already sharing, so we need
Jawar,
to start.
Can you please stop sharing your screen?
Okay, thank you.
So like I said, we're not doing too badly in terms of time. We're only about 9-10 minutes of schedule. And I think considering the number of presentations that we've had, that's not too bad, yes?
Yeah, sure.
So Abey, are you having difficulties?
Yeah, hello. Is there any interruption? Dr. Eric?
Yes.
Any interruption is there?
Yeah.
We cannot see your slides. You can just see your folder with your files.
It's not started.
No. I think you're sharing the wrong window. What we can see is your folder, not the actual presentation.
Yeah, it was started actually. Let me put again. Is it seen? Eric?
No, we cannot. We're still seeing nothing has changed. So probably what you might want to do, just stop sharing and share again, and this time share the correct window if you're sharing a particular window, or just share the entire screen.
Okay. Let me start again.
Because we can see the folder that has your presentation in it, but we are not seeing the actual presentation when we open it. Evanson, do you have Abey's presentation somewhere? We can probably assist him in projecting that, sharing the screen. Evanson or Nelson? Evanson, do you have Abey's presentation?
Okay, let me try one more time if it is open. Is it seen or not?
We're just seeing your folder. We are not seeing your presentation, yes?
Now?
I can see a PowerPoint slide on that screen. It said Dr. Abey, ACP 2021. Can you click on that, please?
Yeah, I already clicked it.
It seems you're just sharing your one window. You're not sharing the actual presentation. Evanson, Nelson, can you step in here and assist?
Did he send his slides?
That's what I'm following up on to see whether we
can
assist.
I already sent slide also.
Oh, there we go. So Abey, that is your presentation?
Yeah.
Okay, so just take us through and they'll move the slides for you. You have 10 minutes, yes? Thank you.
Okay, let's start from first slide. Is it? Hello? May I have previous slide?
Nelson, can you start the presentation from the first slide, please? IT support, Nelson, Evanson, we are waiting on you to share a base slide so that we can proceed. Okay, Abey, you have the floor. You have 10 minutes.
Oh, one more slide is previous one. Okay. Okay, let me start from here. Okay. Is it fine?
That's fine. Yes, let's just make it work.
Okay, so good evening, everyone. I am Prakash Mishra, going to present my research work entitled HPTLC Method Development for Quantitative Determination of Quercetin in Asterium nippellens tubers. Asterium nippellens is a medicinal herb which contains quercetin in its tuberous part. The plant grows in the Himalayan region of India, China, Nepal, and Bhutan. In Uttarakhand, India, it is found at an altitude of 2,400 to 3,000 meters. The plant is used by local inhabitants of Uttarakhand, India as traditional medicine against different types of minor illness. It is also consumed as a tonic as well as in diarrhea, malaria, and dysentery. The extract from the plant's part shows the number of secondary metabolites such as alkaloids, carbohydrates, and flavonoids, etc., etc. So my main aim was to develop high-performance thin layer chromatographic method for the identification and quantification of quercetin in methanol extract of Stearium nepalus tubers. So I am going to describe a simple, sensitive, and accurate HPLC method for the determination of quercetin. In an experimental part, the dried and pulverized 100-gram tuber sample was extracted with 500 ml of methanol. Is it going to so fast, I think? Eric? The previous slide,
Nelson or Evanson, just wait for the speaker to indicate that you need to move to the next slide, please. So previous slide.
Yeah, okay. So in an experimental part, the dried and pulverized 100-gram tuber sample was extracted with 500 ml of methanol by using the hot solvent extraction method for 24 hours and then filtered. The filtrates were collected and the residue extracted twice more at 4-hour interval with the same volume of extractants. The filtrates were cooled and extracted with N-hexane and chloroform with same conditions as methanol. Then all three fractions were cooled separately and solvents were removed by drying with a rotary evaporator at 40 degrees centigrade. All dried extracts were stored in a desiccator for further experiment and analysis. The methanol extract was subjected to sample preparation for HPLC. And the sample was prepared according to 100 mg per ml, and parallelly the TLC plates were kept in oven at 110 degrees centigrade for 20 minutes. The standards were prepared according to the concentration of 1 mg per ml in 50% methanol. And from that, we prepared 6 different standard amounts and were applied on the TLC plate using a semi-automatic Kama Glinomat 5 sample applicator along with the sample. So the next slide. So in this slide, you can see the chromatographic conditions which were used to develop HPLC method. In this method, we used the mobile phase, that is toluene, ethyl acetate, and formic acid in ratio of 7, 5, and 1. The scanning wavelength was 366 nm. Now, next slide. Here is the chromatogram of standard quercetin, and the another one is the sample one, which shows few other peaks, including quercetin. In the table, you can find, oh, it is, please show the previous one. It is skipping, I don't know what is happening.
Previous slide, please.
Yeah. So in the table, you can find all the information about standard sample and calibration. The sample quercetin has an RF value of 0.54, which resembles with the standard RF value. Here, the X denotes concentration of quercetin in the sample according to the height and area, whereas Y denotes regression equation according to the height and area, and the R denotes for the coefficient of the correlation. The next slide, please. Now, method validation. The method was validated for linearity, sensitivity, specificity, precision, and accuracy. In order to determine linearity, a calibration curve was obtained by plotting peak area of a marker compound against the concentration over the range of 2 to 12 microgram per spot. So we can say a good linear relationship was observed among tested concentration. The photo documentation was done in 3 different lights, that is 254 nm, 366 nm, and in white light also, which shows a good resolution of the peaks against the tested concentrations. Next slide, please. So instrumental precision was checked by repeated scanning of the same spot of quercetin, that is 2 microgram per spot, and expressed as the relative standard deviation, that is percent RSD. And the result was found to be 0.71. The sample repeatability was affirmed by analyzing 2 microgram per spot of quercetin individually on the TLC plate and expressed as percent RSD also. The result was 0.74%. The sensitivity of this method was measured in terms of limit of detection and limit of quantification, which was measured using a method based on the standard deviation of the response, and the slope was ascertained by analyzing standard reference and test samples. So you can see the result of LOD and LOQ, that is 0.04 microgram and 0.12 microgram, which indicates an adequate sensitivity of the proposed method. By introducing small changes in the mobile phase composition, such as volume, duration, and saturation of mobile phase, the resultant effects were examined. The robustness of the method was done in triplicate at 2 microgram per spot, and the percent RSD peak area was calculated as shown in the table. Next slide, please. The accuracy of the method was established by performing recovery experiments at 3 different levels using the standard addition method. For that, known amount of standard solutions of quercetin were added to the re-quantified sample solutions of methanol extract. Then their response, that is peak area, was measured and percentage recovery was calculated. Each response was taken as the average of 3 determinations, and the recovery yield for quercetin was found to be between 99.50 and 100.24%. Then the intraday and interday precision of the method were estimated by analyzing 6 replications of 3 samples at different concentration, that is low, mid, and high, containing 2, 8, and 12 microgram per spot of the quercetin respectively, on the same day for the intraday precision and on different days for interday precision. Then the results are expressed as a percentage concerning standard deviation. Next slide, please. Now, conclusion. The method used in this work resulted in good peak shape and enabled good resolution of quercetin from other constituents of the plant material. The accuracy and reproducibility of the method were established in terms of recovery experiment, and the mean recovery, that is 99.81%, was close to the 100%, which evidenced the accuracy of this method. Hence, it is proved to be a simple, reliable, economic, precise, and rapid method for the identification and quantification of this flavonoid in Asterium nipponense tubers. So we can say this method was found to be effective. In fact, quercetin was accurately quantified among all other compounds present in the plant material. So the results obtained showed linear correlation with those obtained from the external references. Therefore, the quercetin can act as a useful biomarker for some chemical studies in the genus Asterium, using it as a standard reference. So we can also use this method for routine quality control of Asterium nipponense tubers, and it can be a good alternative in place of HPLC method, which is more expensive. So this was my research work regarding HPLC method development. So the next slide. So thank you guys for listening me patiently. So I'm sorry for the interruptions.
No, it's fine. We expect some issues whenever we are completely reliant on technology. So thank you very much, and you've taken exactly 10 minutes. So we appreciate that. So,
thank you so much.
Yeah. So, ladies and gentlemen, we've had our 4 presenters from the third portion of today's session. Now, there's a point here, which is that one of the presenters who missed their slot from the previous session is available to present. This is John Mwonjoria. Had some technical difficulties, but he has joined us now.
Well done. Everything is going on fine.
I think someone needs to mute. Okay. So, ladies and gentlemen, if you will allow me some leeway, especially if we are not too pressed for time, we can allow John to make a quick presentation, and then we can proceed to the question and answer session. I find it difficult to turn down someone who was already gone through all the stages to make that presentation. So if there are no objections, or if there are objections, we'd like to hear them. Otherwise, we can allow John 10 minutes to present, and then we can wrap up with one last question and answer session. So John, prepare to present, to share your screen as a way to see whether there are any objections to this. It will take us an additional 10 minutes, but I think for the sake of all of us who stand to benefit from the presentation, that it will be worth it, yes? So again, as a reminder, please take note of your questions. We'll have 5 presenters now to question, so there should be quite a number of questions, either in the chat or you'll have an opportunity to actually ask the questions on the floor. So John, please do not take any more than 10 minutes, yes? Karibu.
Thank you very much for that opportunity. I hope you can hear me, and I hope you can see my presentation.
Yes, we can hear you, and if you can just go to slideshow, probably.
Good.
Okay.
Yes, I'm John Mwonjoria from Kenyatta University. I'd have some co-investigators, that is Kesh Kenyatta and Jomo Miri, and finally we have Jag Elliott. And I would like to present the study we carried out on the antinociceptive and anti-inflammatory effect of ethanol extract of Acidilacta, indica stem, and leaf extracts in animal models. That is a plant I'm talking about. It is also known as the neem in English, or Mwarobaine in Swahili, which means that it can treat around 40 ailments. Now, the part of the introduction, we are more interested with the nociception and anti-inflammatory effect. And the nociception is an indication of the signs that is normally seen in animals when they are experiencing an unpleasant effects associated with the pain. Normally, we call it in pain in humans, but in animals, it's just a perception of what is not very pleasant. And it is normally an important response for the protection of the animal when it's undergoing some injuries. And when we come to inflammation, many of us know that it is associated with the tissue damage, and sometimes it is a bit protective. However, both conditions are associated with the poor quality of life, disease burden, and also they are associated with the high cost of the drugs management, and sometimes even mental illness. And in many parts of and developed part of the world, you find that the drugs that we normally use, that is the non-steroidal anti-inflammatory drugs for the pain, and of course the corticosteroids and other steroids, synthetic steroids, are quite expensive to the people who are earning very little. They are not available because sometimes they have to travel long distances to get this. And even the quality is questionable. However, we have what we call alternative methods. And one of the alternative methods we have in Africa is the use of the medicinal parts or extracts. And one of the medicinal plants used is the A. indica, which is Mwarobaine, as I've just told you. And it has very many uses. It has quite a number of compounds that have been found, that is the alkaloid, the flavonoid, and so forth. And you find that, as I've just told you, much about this plant is not known, but it is believed that it has less adverse effects. it's available, and possibly it can be a source of novel candidates for drug development. Now, our narrow hypothesis was that the stem bark and the leaves extract of the seed plant has no antinociceptive and anti-inflammatory effects. The general objective was to evaluate the antinociceptive and anti-inflammatory effects of the leaves and the stem extract of the neem plant. Specific objective was to determine the effects of the leaf extract and stem extract of the plant on formalin-induced nociception in rats, on teofric at the hot plate test in mice, and the formalin-induced poedema in mice as a model of acute inflammation. Methodology: the plants were collected in Mombasa during the day. They were brought to Kenyatta University. They were dried, the shade away from sunlight. They were ground, and they also identified in the herbarium. They were ground in powder. They were extracted with an ethanol, filtered, and then concentrated using a water evaporator at reduced pressure. Then stored in cool dry place for future use. And antinociceptive assay, the animals that were used were the white winged rats and the Swiss albino mice. The white winged rats were used for formalin test. The Swiss albino mice were used for teofric and hot plate. They were divided into 6 groups, and the number of in each group was 5. The various treatment, the various doses of the extract were, and also the controls were administered peritoneally. Pain was induced, formalin pain was induced by injecting 5 mils of 5% formalin in the subfrontal region of the head pole. And the pain was quantified by measuring the time spent in lifting, licking, biting, friction, and the friction of the affected pole. It was measured in two blocks. One block was 0 to 5 minutes immediately, and that was the acute pain. Then there was a chronic pain, which is the second pain, between 15 and 30 minutes after injecting with formalin. The hot plate test, the animals were placed on a hot plate, which was heated up to the 50 degrees centigrade, plus minus 1 degree centigrade. And the time taken by the animal to jump or even lift the leaves was quantified as the latency of nociception. And a maximum of 20 minutes was set, so that beyond there, the animal was removed. Now, the teofric test that was carried out was the immersion one, whereby the tail was immersed in a beaker which contained water, which was heated to 50 plus minus 1 degree centigrade. At the time taken for the animal to freeze off the tail was noted, and it was taken as the latency of nociception. Anti-inflammatory assay, the animals used to here were the Swiss albino mice, again, the same grouping, and the treatment was the same. And the inflammation was induced by injection of 5 mils of 5% formalin, again in the subfrontal region of the head pole. The pole diameter was determined using a digital calipers, and the difference in diameter was measured every 30 minutes for 2 hours. And the difference from the initial one was taken to be the edema developed in the head pole. Data analysis, the data was expressed as means of the mean.
John, yes? if I can interrupt you, have 2 minutes to finish up. Yes, 2 minutes.
Okay. Yes. The data was expressed as standard errors of the mean, and then analyzed using one-way ANOVA at the CIPS post-hoc test. And significance was set at P being less than 0.05. The result and discussion: the leaves extract or the leaves extract, the stem extract of the plant exhibited significant antinociceptive effects, as indicated by the stars. It shows that P is less than 0.05. The same was observed with the stem extract here. And when we come to the theophoric, again, the diclofenac, which is a standard at 50 milligrams, exhibited significant antinoseptic effect. The same thing was observed when you come to the hot plates with the stem, whereby again the 50 milligram exhibited significant antinoseptic effect. And the same also was seen with the leaves when it comes to theophoric. Again, the 50 milligram is tending the same. But when we come to the leaves, the theophoric, you find that both 12.5 and 25 milligrams exhibited antinoseptic effect. But when you come to the anti-inflammatory effect, we have a different scenario here whereby the 12.5 milligrams and the 25 milligrams exhibited anti-inflammatory effect, while the diclofenac, which was the standard drug, exhibited anti-inflammatory effect on the 150th hour. In conclusion, the extract attenuated the nociception in both rat and mice and inflammation in mice. And this may indicate that we have phytochemicals and secondary metabolites with antinoseptic effects, and it supports the focal use of the plants in pain and inflammation management. And with more studies on the plants, it's a potential, it may be a potential source of new raw materials for the manufacture of analgesics in future. Thank you.
All right. So thank you very much, John. And thank you very much to all our presenters. And thank you very much to all our participants for hanging in there. It's been a long day of a lot of science. And so what we are remaining to do now is to have a quick question and answer session for the 5 presenters. Actually ask your questions on the floor. So questions and answer session is now open. I can see one of the questions that was typed in here. This question is for you, Jawar. I hope you're still with us. So the question is, what were the polyphenols that you screened for and the antioxidant effects of the same? So Jawar. I think he might have left. No, he's still with us. Jawar, if you can unmute. Okay, I think he probably may have stepped out. So this question, the next question is for Mohammed. I'm guessing Mohammed Salisu, if I'm not wrong. So the question is, why did you choose the models that you used for your research work? So can you provide a brief justification for the models that you used for your work? Mohammed?
Thank you for that wonderful question. Can you hear me?
Yes, we can hear you. Please go on.
Thank you very much for that wonderful question. I choose that model because they are actually models that are actually used in central and peripheral analgesic activity of a particular organism. Actually, those models are the models that are used for checking the ability of a particular extract in terms of knowing the central and peripheral analgesic activity of a particular plant.
Okay, and there's another question still for you now that you're on the floor, about why you did not, or whether you did perform any quantitative analysis of your extracts.
Quantitative?
quantitative, yes.
Quantitative analysis were done actually. We in the table in the slides, table 2 showed the quantitative analysis of the phytochemicals actually, where we have the alkaloids to be the one of the highest yielded phytochemicals presence in that extract.
I'm guessing possibly the ask of
the person probably he didn't follow up when I was doing the presentation.
Or maybe they were waiting to see some statistics, ED50s, IC50s, and so on and so forth. Yes.
Okay.
Possibly that's where they. Okay. Thank you very much, Mohammed. Fatima, I hope you're still with us. There's a question for you. So the question is whether or not what were the phytochemicals that are present in the plants that you worked on? Whether there was any phytochemical screening? Yes.
Yes. Okay. For this study, we didn't do it, but we did an initial study before this. So there were many phytochemicals. We had the alkaloids, the tannins, saponins. We had the cardiac glycoside and also triterpenes. But for the course of this study, yes. But this is a further study on that. That was why we didn't present phytochemicals.
Did you, at the very least, in try and do some fractionation of those extracts that you are working on?
No. Yes, I have the N-hexane fraction. Initially, we did the acute toxicity, determined doses, and then the phytochemical screening. And so far, we have carried out an analgesic and anti-inflammatory studies on the N-hexane fraction. So we're still working on it. It's still a work in progress.
On other fractions, because the mechanisms that you've looked at are quite specific, yes. And so we'd want to see you testing simpler mixtures, if not pure compounds, yes.
This is actually a continuation of the study.
Excellent. So what you're telling us is that, again, we should look out for publications coming from you in the
near
future.
Yes, sir.
Exactly. Thank you very much. Okay. So moving on, there's still another question here for you, Fatima, whether this is a quick one, whether or not you used hot or cold maceration in your extraction.
Okay, I used a cold maceration method.
The cold maceration extraction, yes? Okay. So they're still with you, Fatima. There seems to be a lot of interest in your work.
My work, okay.
Yes, someone is saying, I saw streptomycin as part of your drugs, but it wasn't used in the work. Can you explain? Well, did you mention streptomycin?
I didn't get
and
you.
What was it? Did you mention the drug
streptomycin? Yes. You know, it was a cotton pellet induced granuloma method. So in the course of implanting the pellets, it's possible they might develop sepsis or septicemia. So to prevent infection, we used this streptomycin and penicillin. It was the solution was made using distilled water, 0.2 ml, streptomycin 0.13 milligram, and penicillin. So the pellets were soaked before the implantation. The autoclaved pellets were soaked in this mixture and then implanted in the groin of the rats.
Okay, so the main role of streptomycin there is to prevent infection.
Is to prevent infections.
Okay, excellent. Back to you, Mohammed Salisu. There's a question here on this choice of methodology. So the question basically is quite long, but it basically reads, why didn't you actually consider determining actual LD50s? This is for your acute toxicity, yes? So to determine and report actual LD50s instead of just stating that they are in excess of 500 milligrams per kilogram. What is your response, Salisu?
Well, thank you very much for this wonderful question. Actually, in this study, we both carried out the methods, two methods in analyzing our LD50. We did that of the LOX method, 1983, and that of the OECD 2008, using up and down method. Actually, we did the LOX method first, then we reaffirmed we confirmed the result obtained from the LOX method using the OECD guidelines. Okay. So we didn't actually did only OECD. We did the bot method.
Okay, so it wasn't an oversight. This is because the LD50s are actually very high. Yes, very little toxicity. Yes.
Yes.
Okay. All right. Now, still with you. There's another more general question here on why you opted to do 2 rodent models. What particular value was there in using both rats and mice for your toxicity evaluation?
You know, actually, sir, in these models, actually, we wanted to see the best method that best, particularly to a certain animal models, like the formalin-induced pack killed test is mostly very open and seen clearly in the using the rats instead of the mice. So that's what's involved coming up with the both models. Because, and the acetic acid induced and the hot plates are also very seen clearly also using the mice model.
So in your recommendation, someone wanting to carry your work forward, which of the 2 rodent models would you recommend that they continue with? The rats?
Yeah, it all depends on the methodology that you want to actually apply. And like I said, the formalin is more clearly seen in using the rats instead of the mice.
Okay.
Yes.
Thank you very much. Fatima, back to you. Like I said, your work seems to be generating quite a lot of interest.
Yes, sir.
So you're being asked to explain why you think 250 milligram per kilogram dose of your extracts gave a high activity compared to the 1,000 milligram per kilogram dose.
Actually, it was on, when I saw the question, I went back to check. I saw it was on GSH. What I think maybe the reason is that, okay, maybe at the lowest dose, that means the extract is capable of increasing GSH as the very lowest dose. Maybe by increase in dose might not increase activity. That's the only thing I think I can say about it. I don't know if anybody can help me out.
Okay, so
that's what I think. That means at the low, very low dose, the extract is capable of increasing the GSH. Yeah.
Okay, so this is not
that's increase in dose. That's doubling the dose will not increase the potency.
Okay, you lose that dose concentration response relationship. So this is not the first time you're seeing responses that are varying with concentrations. Yes. Okay. All right. Thank you. But that's probably something that needs to be looked at. Now, we're moving now to John. John, who's just made his presentation. So you're being asked, why did you use the formalin-induced poedema model rather than the carrageenan-induced model for your work? Any response to that, John?
Yes, thank you. There are several methods that you can use in inducing edema in rodents. One of them is carrageenan, which is very good. However, formalin is also quite good. We used formalin on this case because the carrageenan we had at that particular time had some issues. It had been important and then it was kept banned, and the experiment had started, and it required that we had to import in another one. And much of this work was done by my students. And because of the shortage of time, we opted to use formalin, which is as good. In fact, it gives very good, robust results. The only shortcoming in the formalin is that it also causes a lot of pain to the animal, a lot of suffering. That is the negative part of formalin. However, it was managed due to cost.
Okay. All right. So it was not an oversight. Thank you. Still with you, John. There's a question here, probably on some follow-up work. Why didn't you go on to measure inflammatory markers as part of your work, now that you're looking at anti-inflammatory effects?
Actually, this is an ongoing work. The next level will be to fractionate, to alkaloids and the flaavonoids, and then find out what is happening, and then from there we look at the mechanism.
Okay. All right. So that's ongoing work. So again, we've had this once or twice before that we need to look out for for publications coming from you on the follow-up work. Excellent, excellent. So this session is warming up nicely. There's another question that has just come in for you again, John, before you mute yourself. How would you classify your extract based on your results? Is it centrally acting? Is it peripherally acting? So a quick comment, John.
It is both peripherally acting, because from formalin, we saw an activity from the first phase, and also from the inflammatory bit, which is the second pain. And when you come to the what we call the dermal pain, that is the hot plate and theophoric, the theophoric shows that it is a supraspinal. It is spinal, while the hot plate is supraspinal. Therefore, it means it is acting at the level of the spinal cord as well as the brain, and also at the periphery. So it is all over. So I think it's a very interesting study. It ought to be taken by several people to continue if you are going to actually. It appears to be a wider area that requires to be investigated by many.
Yes,
I would invite collaboration.
Exactly. Mechanistic investigations there seem to be the next step. Yes. Excellent. Now, I don't know whether Jawar is back.
Yes,
Jawar,
yes.
Are you with us?
I'm with you.
There was a question for you at the very start. Probably you can ask it again. So this had to do with, did you look at specific polyphenols? Did you screen for specific polyphenols as part of your work for the antioxidant effects?
Please repeat. What?
So the question here reads, what were the polyphenols that you screened for as part of the antioxidant activity that you looked at?
Flavonoids, flavonol, and condensed tannins.
Okay.
Tannins.
All right. So, and I have one more question, just a follow-up to your presentation as well. So, you are looking at the extracts of this plant, yes? Did you do any fractionation of those extracts, trying to simplify the mixtures and redo the assays?
No. We just did the screening, the chemical screening as a first steps to do other phytochemical assessments.
Okay, so that is
it was the goal after all the activities that we found in liver and kidney. So just a preliminary work on polyphenol, total polyphenol, and we found that flavonoids represent a high percentage of these polyphenols. So flavonoids are well known as antioxidants, strong antioxidant molecules. So the step, following step is to fractionate. Yes, and to search for exact structures of these molecules.
And I hope that is work that you intend to carry out going forward, rather than leave it as it is. Yes? Is
what?
Ongoing work? I'm asking whether this is ongoing work?
Not yet. We're searching for yes.
Yes. Okay.
Because yes. We are searching for a group, chemical group. We have not. I'm new in the Faculty of Medicine of Tangier, donc so we are searching for a team that will continue that can do, and the doctor and also a PhD student that can be involved in this work. So inshallah, it will be in the future.
That's quite reasonable. Thank you very much.
Near future, I wish. Thank you.
All right. So one final question to you, Salisu, as we wrap up this session. I think we've had a very informative session. So do you plan on assessing the effects of your extracts?
Yes. Thank you very much for this wonderful question. This is an ongoing research. Actually, we intend to do that, even though that aspect has not yet been been started.
Okay.
Yes.
All right, but the intention is there, and that's where it
Yes,
starts.
we intend to do that. Yes.
Excellent. So, ladies and gentlemen, floor is still open for probably one final question as we wrap up this question and answer session. It's been an exciting natural product session, right from our lead speaker and other speakers who followed. Excellent range of presentations, pharmacological investigations, method development, and I think it was well worth the wait for these particular sessions. So as I give room for probably one or two final questions, I need to make an announcement. I think you probably have seen it in your chats, because what follows from this session is the poster presentation. And as you recall, one of the things that people were asking for was the link to the poster gallery that has been provided in the chat. So you can all find that link in the chat, and when you follow that link, it will take you to the gallery, the poster gallery. The instructions are there on the poster gallery, and it's quite intuitive to follow on from there. Yes. Right. So seeing that there are no more questions, and I think we've had a very long session, it remains for me to thank all of you, to thank all our speakers for the excellent presentations, to thank the members for engaging the speakers and asking very many questions and showing an interest in the work that has been presented here. It has been my pleasure to guide us all through this particular session, and I look forward, like I said earlier, we have 2 more natural product sessions. Yes, we have another session tomorrow, and we have the 3rd session on Friday. There is a lot of interest in natural products. So I'm hoping that we shall all again meet at the next natural product session tomorrow. So right now, it remains for me to thank you once again and to wish you a good evening, even as we proceed to the poster presentations. Now, in case you have any issues navigating through the poster gallery, you can always return to the Zoom main room, not these breakout rooms, but the main meeting. And there the ICT personnel are waiting to handle any of your queries. So should you have any issues, you can always come back and ask. So enjoy the poster gallery presentations, and I look forward to seeing you again tomorrow. So thank you very much from me, and we can now depart from this breakout room. Thank you very much.